Okay, our final presentation of this afternoon's session is from Jose Bezagato from the University of Londrina in Brazil. And the title of this presentation is Changes in the Fecal Microbiota of Beef Cattle Caused by Confinement and the Use of Virginiamycin as Growth Promoter.
Hi, I'm actually Marcio Costa, the last author. Bezagato is one of our students. So thank you very much for hanging there for this last presentation. Many of you may have heard that we carry more bacteria in our bodies than human cells, and that those bugs are very important to health maintenance, as disruption of this balance may lead to, for example, obesity, diabetes, or even behavior changes like depression. So we only know that is already very well established because of the development of new sequencing technologies. In the past, we used culture to address bacteria, but only 20% of bacteria really grows in those media. So now we use DNA sequencing to better address those changes. And many studies have been done in the bovine over the last years. But most of them address the changes in the ruminal bacteria deemed to be more important, and they certainly are, but that doesn't mean distal gut bacteria aren't important. So here we are trying to figure and to understand a little bit better what changes in management by confinement of those beef cattle cause in the distal gut bacteria. Also, what is the impact of the use of Virginiamycin as a growth promoter in those animals? In order to do that and to answer that, we used 30 crossbred beef cattle that were arriving to a feedlot unit down in Brazil. And we divided those in two groups, one receiving Virginiamycin and a control group receiving the same diet without the antibiotic. This diet was, there was an adaptation period over a week in which we gradually increased the amount of carbohydrate to 60% in a dry matter basis. And we collect samples from arrival and again 25 days after and at the exit to the slaughtering. And basically, we extract the DNA from the feces, and then we amplify the V4 region of the 16S by PCR, which is a gene specific for bacteria. And we sequence that using Illumina technology and compare those sequences with a public database just to obtain some taxonomic information. Classification. And then we did a bunch of statistical analysis to compare samples between them. But before sharing my results with you, I would like to review two basic concepts. The first one is composition. Composition means which bacteria are there. So in this example, we see that there are two communities that are comprised by 5 species of bacteria, so they have the same composition, right? A and B. But when we consider the distribution of those species, then we are talking about structure. And in this example, they have the same composition, but you can see that they have completely different structures, right? And here are our results. Considering composition of the bacterial communities in the feces of those animals, we did have a cluster. So this analysis basically puts together similar communities, okay? So samples that had very similar communities, they are close to each other. And here we see a nice cluster of animals arriving. So all the samples collected arrival cluster together, and makes sense since they were in a pasture-based diet. And thanks God there was no difference between antibiotics and control because they were not treated at that time. And 25 days later, we see that the bacterial community completely changed. And we deemed that to changes in diet, as it has been previously showed. But treatment with virginiamycin did had a big impact on those, and the composition of those communities. That means that the antibiotic is selecting for certain species of bacteria, and this effect was very consistent among all the samples of treated animals. And what was also interesting is that at exit, 90 days after entrance, there was no difference between antibiotic-treated animals and controls. We are not sure why that would happen, but we believe that adaptation of this microbiota or metabolization from these drugs in the ruminal bacteria, but this deserves more investigations, further investigations. And when we talk about structure, that's what we found, kind of similar results. Samples from arrival, they all clustered together. No difference between treated animals and controls. And 25 days later, we see that there was formation of 2 clusters. And antibiotic-treated animals, they also have a very similar structure in their communities. Okay, I have to disclose that depending on the statistical tests that we apply here, they are not statistically different, controls versus treated groups. And we believe that's because of that outlier animals that are on the top. But it is very clear that you can see the formation and the grouping of those treated animals all together. And again, no difference between controls and treated animals that exit from feedlot. Oops, I think this is the old version of the presentation. Okay. And you might be thinking, so why is this important? And for example, in this analysis, I won't have time to share all the results of our research, but in this analysis, we compared the fecal bacteria from the animals that had the highest average daily gain with the animals that had the poorest weight gain. And here we can identify different genera of bacteria that were more likely to have a biological significance in animals with higher weight gain. So the directions that we should follow is that, like they are doing in humans now, microbiota manipulation, all right? So if we can develop a method to favor growth of those bacteria present in animals gaining more weight, or disencourage or decrease the growth of those bacteria that might not be very efficient in breaking food, and therefore they don't yield much energy from the same feeding, we may ultimately be able to replace or decrease the use of antibiotics in our management systems. So, in conclusion, animals entering into the feedlot, feedlot had a big impact on both composition and structure of their intestinal bacteria, and also did virginiamycin, but we need more studies because it seems that virginiamycin has a bigger impact on composition of those bacterial communities in those animals. I would like to thank the Brazilian funding agents for supporting this research and my colleagues and co-authors who collaborated with me, and I'm happy to answer any questions you may have.
Okay. Thank you very much, Marcio. So again, we do have time for some questions, so I'm very happy to open this paper to the floor. So, yes, we have a question at the front here. If we could get a microphone to the front, please.
Have you been able to correlate different families with different health events in those animals? In other words, what's the healthiest microbiome to have?
Yes, not in this research. None of the animals got sick, so we couldn't really. There is a bunch of studies being done with ruminal acidosis, but not for these animals. We didn't go very high with the concentrate ratio, so we stopped at 6%. That was a commercial farm. That's what they were doing. We just didn't want to do anything different. So we were there and collecting samples. So none of the animals had laminitis or acidosis or anything like that.
Okay, thank you. Do we have any other questions? Yes, we have a question here. Behind you, if you want to.
I was just interested in, if we're going to use, Bob Reinberger, Australia. I was just interested in the use of the Virginiamycin, because we do use it on introducing animals onto feedlots and even onto dairy cattle when they come into to milk. I was just wondering if what we do is we normally then reduce that as we go along. Is it a possibility that all we're doing is when we put the animal into the feedlot, the new bacteria are actually growing a lot faster, so the lactobacilli and the material that's making it more acid in the rumen, and the reason that then it settles down is because they become more used to that new diet, and all it does is the Virginiamycin just allows that to flow along a lot simpler than the ones that we don't use the Virginiamycin in.
Yes, that's a very good question. So I'm gonna, I had another slide here, but I'm presenting a poster, being 25, here at the same floor, in which we collect samples from the rumen of those same animals at slaughter. And Virginiamycin still has an impact on the bacteria in the rumen at the time of exit. You don't see that effect on the, thank you. We don't see that effect, this presentation was, here. There we go. There is still a difference, but that's a good question. I don't know what's really the importance of this at the time of slaughter. You know, what I want to do with this research is to identify taxa, as you can see in the figure 3, in which we can replace the use of antibiotics, just causing the same effect that the antibiotic will have in this microbiota, so these animals will gain more weight. So that would be.
Okay. It sounds like you may have other things that you can discuss with the person who has the question for you at a later point. So again, I'd like to thank Marcio again for a very interesting presentation.
Thank you.
And, that's the final presentation of this afternoon session on nutrition. I'd like to thank you very much for your participation in the session. So the session is now closed.